All right, yeah, so thanks for having me here today. My name is Johannes Linder, and I'm a PhD student from Seekprop Lab at UW. And the title of my talk is Predicting, Engineering, and Interpreting Biological Sequences with Deep Learning. And feel free to interrupt me with questions if things doesn't make sense, or if you want me to explain things a bit more in depth. So, the human genome contains more than 3.1 billion base pairs. And the vast majority of these nucleotides do not code for a particular protein. Yet genetic variation in these non-protein coding regions can have a profound impact on gene expression. And the reason, of course, is that there's a complex cis-regulatory code hidden in the genome that, in a sense, governs gene expression. So today, I'm going to talk about 3 challenging problems related to the non-coding part of the genome. The first problem is that of variation. So genetic variants in these non-protein coding regions have been found to be deleterious and causal or contributing to genetic disorders. Then, of course, it's important that we develop models and methods that can infer the impact of mutations in the genome and pinpoint the ones that are causal for a particular disease. The second problem is that of rational sequence design. So here, the goal is to design improved gene regulatory sequences and proteins for some target biological function. Now, the applications of rational sequence design are, nearly endless. If we can make improved gene regulatory sequences, we can design better drug delivery systems and vaccines. And if we can make better proteins, we can design improved antibodies, enzymes, and other therapeutics. And the final problem that I'm going to talk about is sequence interpretation. Okay, so let's say now that we have these accurate models for pinpointing the genetic variants that seem to be disruptive and causal for some disorder or phenotype. That's usually not enough, right? We probably need a very detailed understanding at the molecular level of mechanistically how these variants are disrupting normal gene expression in order to take action and combat these variants, for example, by developing some therapeutic. Then, of course, it's crucial that we develop models that allows us to, or methods for interpreting any model that we build. So the first problem that I'm going to talk about, variant prediction, in this context, I will present a neural network that we developed in the lab for predicting an important RNA-regulatory process. So let's jump into that. So this model is called a parent or APA regression network, which is a deep neural network that we trained on a very large set of massively parallel reporter assay data to predict an RNA process called Alternative Polyadenylation. And this work was published in Cell a while ago. So polyadenylation is a process that occurs usually in the 3' untranslated region at the far end of all human mRNA molecule, where a large protein complex, usually referred to as the 3' end processing machinery, recognizes a specific sequence regulatory code that we call the polyA signal, that's here illustrated as the dark green box. And this protein complex cleaves the RNA molecule within this signal and appends a long string of A's, A-tail. Now, alternative polyadenylation is the phenomenon where you have multiple competing polyA signals in the same RNA molecule that, of course, gives rise to competing isoforms. And in fact, more than 50% of human genes have more than one polyA signal. And these different isoforms typically have very differential effects on, let's say, stability. And the isoform abundance between these competing polyA signals depends on to a very large extent on the cis-regulatory code of each polyA signal. So then the question we asked is whether we can predict the isoform proportion between 2 competing polyA signals, as well as the cleavage distribution across an input polyA signal sequence, given only the sequence information alone, because that would give us a very powerful tool to predict variants in the genome. And so to that end, we developed this neural network called a parent that accurately predicts the cleavage distribution at base pair resolution. So the training data for a parent was created by a very talented individual, Nick Bogard, that used to be a postdoc in Seelig Lab. He created this very large massively parallel reporter assay, where he essentially took a native human 3' UTR and that natively included 2 polyA signals, and replaced the proximal polyA signal with essentially fully randomized sequence, and then transfected and grew these plasmid reporter gene mutants in a human cancer cell line, HEK cells, and used next-gen RNA sequencing to measure the isoform proportion and cleavage distribution of each degenerate polyA signal. And this resulted in a remarkably large and diverse data set with 3.5 million randomized polyA signals with associated measured cleavage distributions at base pair resolution. So given this data, we trained a multitask convolutional neural network to predict APA. So this network takes as input a one-hot coded representation of the proximal polyA signal. The first convolutional layer consists of short 8 nucleotide wide filters that learn to recognize short important determinants within the sequence. The second convolutional layer has a larger net receptive field across the input and can learn to detect longer motifs, repeats, or combinations locally. These convolutions are then fed into a dense, fully connected layer that finally is passed to either this isoform prediction layer or this cleavage distribution prediction layer. The isoform predictions are handled by a simple sigmoid activation, and the cleavage distribution is modeled by a softmax categorical distribution with independent regression weights. To train this network, we jointly minimize the isoform proportion and cleavage distribution KL divergences with respect to our training data. And after training, we tested a parent's ability to infer the isoform proportion of held out test sequences, randomized test sequences. And as we can see, the predicted isoform proportions correlate very strongly with the measured isoform proportions. Furthermore, and importantly, in addition to these randomized polyA signals, Nick Bogard also measured the isoform proportion of native human polyA signals in the same APA reporter system. And this is quite cool, I think, because a parent that has only been trained on these degenerate polyA signals can accurately infer the isoform proportion of human polyA signal, which of course is a requirement if we want to do variant prediction. And I think this is also a quite nice visualization of how powerful a parent is. So here we're looking at a sample of 3 signals from the test set. And what I'm plotting as a dotted curve across each polyA signal sequence is the measured 3' cleavage distribution. And as you can see, the predicted cleavage distributions of a parent agree really well with these observed distributions. So after this, we wanted to get an understanding of what exactly a parent had learned in its internal network architecture and whether we could extract useful biology from this network. So to start with, we employed a visualization technique that had previously been developed by another research group, where we essentially study each filter in isolation. So here, we're searching for subsequences. We try to find a representative set of subsequences from our data that result in maximal layer 1 filter activation for each filter separately. And we try to get a large representative set of subsequences that maximizes the filter activation that we can then stack into a position weight matrix such that we get a sequence logo that represents the consensus sequence of each filter. And to estimate the positional effects of each filter, we performed simple Pearson R correlation testing between the activation maps of each filter and the predicted outputs. And when we compare these sequence logos to TomTom, the TomTom database, we find that many of these filters match known RNA binding protein motifs, and the estimated positional effects of each filter across the polyase signal sequence agree quite well with established biology. We then extended this same visualization technique quite seamlessly to the second convolutional layer. So now we're searching for longer subsequences that ultimately result in maximal layer 2 filter activations. Of course, the subsequences are now longer because the layer 2 filters have a larger net receptive field across the original input sequence. And what we found here is that these filters typically match combinations of short RNA binding protein motifs, which is quite nice because it sort of indicates that the second convolutional layer learns to be sensitive towards combinations of or local interactions of individual APA regulators. So now when we have this accurate model of APA, we wondered whether we could use this as a tool to infer the impact of APA variants in the genome. So given a wild-type polyase signal sequence and a variant sequence, we can individually score these two signals using the apparent model. And as a proxy for the impact of the mutation, we can estimate that as the odds ratio of our predicted isoform proportions of the variant to wild-type sequence. So to test apparent, we collected human variant data from the ClinVar and HDMD databases, and we also performed saturation mutagenesis of the 3' UTRs of ACMG genes. And Nick Bogard went back to the lab and experimentally measured both the wild-type and variant sequences of these variants in order to measure the fold change of each variant. So in this plot, we're plotting these more than 12,000 measured variants. On the y-axis, we see the measured observed fold change of each mutation. On the x-axis, we plot the relative position of the mutation with respect to the central hexamer, which is the core element that defines a polyase signal. And then the color corresponds to the apparent predicted fold changes. Now, as you can see, the measured fold changes along the y-axis correlate very strongly with the apparent predicted fold changes. In other words, the apparent predictions are quite accurate. And finally, this is not published work, but recently we kind of reevaluated how well apparent can score variants. And in particular, we benchmarked its performance at the task of classifying disruptive APA variants on our data and compared it to other sequence predictive models of APA that have come out either afterwards or during our initial publication. And so here we're looking at the more complex task of classifying disruptive variants outside of this central hexamer, because variants occurring across the central hexamer are quite trivial to classify. So now we're looking at this set of more cryptic APA variants. And when we compare the precision recall curves for classifying these variants, we see that apparent, the orange curve, has a significant advantage in predicting these variants. And I don't really have to show more of these results, but we sort of validated also with independent data sets or MPRAs from other groups that these trends generally seem to hold. And in also unpublished work, we recently developed an improved apparent model that is based on residual neural networks for predicting APA. Very similar to this very cool BPNet paper that recently came out. And so here we replaced this sort of classical 2 convolutional layer architecture with a very much deeper neural network consisting of 28 residual blocks stacked in sequence, where each residual block contains 2 convolutions and a skip connection. And this network finally predicts the 3' cleavage distribution at the end. So it essentially transforms the input polyase signal into this 3' cleavage distribution signal. And if we then reevaluate the models on classifying disruptive variants, we can see that this residual network apparent model is significantly better than the original apparent model at the task of classifying these human variants. And of course, this is without any fine-tuning on that variant data. Again, we're training only on these randomized poly-A signals. Which, I think this is really cool because it essentially shows us that, as we get better and better at machine learning and deep learning, we get better results. So now let's switch gears and jump into methods for designing de novo gene regulatory sequences and proteins based on these sequence predictive models as a guide. So the goal in computational sequence design is to generate sequences with improved biological function, given some initial measured data. And the running example that I'm going to use in this presentation is to engineer improved polyadenylation sequences. And the quick motivation for that is that it's quite well known by now that efficient transcription termination is associated with stronger net gene expression. And so if we want to design gene constructs or gene circuits that have very strong gene expression, we may need to engineer maximally strong poly-A signals, and potentially in the future even engineer cell type specific poly-A signals. So generally speaking, if we want to design biological sequences, we can turn to these classical discrete optimization methods, such as genetic algorithms or simulated annealing, which is a form of MCMC. However, it's quite well known that these methods sometimes are not very sample efficient, meaning that they require many calls to the fitness predictor model in order to converge for a single sequence design. And this, of course, matters. Speed matters if we want to design many sequences, or for that matter, if we want to design many sequences for many objectives. As an example, let's say that we wanted to use this to design a large representative set of sequences that maximizes every neuron in our neural network as a form of visualization. Then, of course, we're going to be generating potentially even millions of sequences. And then speed really matters for most of us that don't have like a TPU farm or anything. So here I'm going to talk about a quite simple heuristic method that we developed that seems to be very fast at designing sequences that we call Seekprop, that is based on the simple idea of gradient ascent or activation maximization, as it's also called, with some added modifications that makes this suitable for sequence design. So in fact, gradient-based input optimization was previously proposed, had previously been proposed, where the idea is essentially to optimize a softmax relaxed representation of a sequence, also called a position weight matrix or PWM, using gradient descent. So in this scheme, we start with a matrix that has the same shape as a one-hot coded sequence, but that consists of real valued optimizable parameters. These nucleotide logits or parameters are transformed with a softmax across the columns to get our softmax sequence or PWM. PWM is then directly fed as input to the, allowing us to evaluate our differentiable fitness objective. Sorry, it warns that my connection is a bit unstable. Hopefully you can hear me. Otherwise, feel free to tell me if I'm talking to no one anymore. All right. And then we can essentially use backpropagation because this whole pipeline is differentiable to optimize the parameters to maximize the predicted fitness. Now, what we noticed was that this original version of the method converges quite slow in practice. And more importantly, this fitness predictor model has only ever been trained on proper one-hot coded discrete patterns and may perform arbitrarily bad on these continuous relaxations. So we developed this method that we call Seekprop that makes some technical modifications to make the activation maximization more suitable for biological sequences. And the latest version of this method we call fast Seekprop because it's faster. And so again, we start with these optimizable nucleotide logits, but before transforming them with a softmax, we start by applying a parameter normalization layer that keeps these nucleotide logits zero-centered and proportionally scaled across positions. And furthermore, this normalization layer essentially introduces a global temperature parameter that controls the total conservation of our PWM. And what's really nice is that this temperature parameter is actually also trained with gradient descent, which is nice because it removes that hyperparameter for us. And this seems to result in quite a significant speed up. Secondly, instead of feeding the softmax sequence directly to the predictor, we can insert a discrete stochastic sequence sampling layer, where we now pull discrete sequence samples and feed it to the predictor model. This avoids sending these bad continuous relaxation predictor. And to backprop, we rely on a gradient estimator, such as the Gumbel distribution or straight-through approximation. And here, I'm showing you the results of generating poly-A signals with maximal predicted isoform proportion according to the apparent predictor model. And in the plot I'm showing on the right here, we're comparing 4 different design methods where they're all initialized with some random start sequence. And then the x-axis corresponds to the number of changes we make to the sequence, or alternatively, the number of predictor calls we make. And the y-axis corresponds to the predicted fitness score of a parent. So as you can see, the purple trajectory for fast Seekprop converges to higher fitness scores considerably faster than a method such as simulated annealing, which is the black curve. And by comparing these results on another model, so a separate model that we did not explicitly optimize these sequences for, we can sort of validate that our design sequences do not, in a sense, overfit to the apparent predictor as sort of a sanity check. And so here we're comparing our designs, these trajectories, according to a independently trained model, D-Rate APA. And as you can see, these trends seem to hold up in that the fast Seekprop sequences converges to very high validation scores faster than the other methods. And finally, I want to show how we can experimentally validate our sequence designs. So here, we try to generate poly-A signals according to target isoform proportions. And we also generate poly-A signals with maximal isoform proportion. And also, this experiment is a bit older than our latest version of this design method. So here we use the original Seekprop method that doesn't use any normalization. So it's a bit worse and slower than the latest method. But yeah, we haven't experimentally validated our latest designs. But so here, Nick Bogard was convinced to, again, go back to the lab and synthesize and express these designs in a human cell line and use RNA-seq to measure them. And as a result, we generated and measured more than 1,200 poly-A signal sequences. And what I think is really cool is that the measured observed isoform proportion of these designs correlated very strongly with the target isoform proportions that we set out to design them for. And the sequences that were designed to be maximally strong were in fact stronger than any native human poly-A signal that we had measured in our APA reporter system previously. So that's great, but what if we want to scale up even further? So what if we want to now generate millions of optimized diverse candidate sequences that we can then subject to high throughput screening in an MPRA, in a wet lab setting? If we want to do that, then activation maximization is, of course, terribly inefficient because we have to rerun activation maximization millions of times without even any guarantees that we're going to end up with diverse designs in the end. So instead here, what we want to do is that we want to train a parametric generative model that allows us to sample high fitness sequences with high probability. And so here I'm going to talk about an extension that we developed based on activation maximization that turns it into a family of generative models where we use backpropagation to train this generator to sample diverse optimized sequences. And this was published in Cell Systems a while back. So the basic idea is to rely on backpropagation to jointly maximize both the predicted fitness and the expected diversity of this generator based on Monte Carlo samples. So the trick is to independently sample 2 generator seeds that we feed into the generator separately. And as a result, it returns 2 independent generated sequence samples from its model distribution. We can take one of these sequence samples and feed it to our fitness predictor in order to train the generator to produce maximally fit sequences. And simultaneously, we can take these 2 Monte Carlo sequence samples and compare them on the basis of some differentiable similarity comparator in order to train the generator to produce maximally diverse designs. Now, this similarity comparator can be something simple, like the cosine distance among the one-hot coded representations, or it can be something more complicated, like the latent similarity in some autoencoder feature space. Optionally, if our predictor model has regions of epistemic uncertainty, we may also need to maintain confidence in our sequence designs as we're designing them. To that end, we can hook up a differentiable likelihood model, such as a variational autoencoder or an autoregressive network, to maximize the likelihood of our designs with respect to the training data. Now, this system is not perfect, and I guess one of the bigger caveats here is that this compound cost function naturally has these nuisance hyperparameters of how much we weigh each cost term that are quite hard, if in some sense not quite impossible to set optimally. In our experiments, we sort of went by some simple heuristics that in the end produced quite good results empirically. So in our experiments, we tuned this diversity weight such that the predicted fitness of our generated sequences approached as well as possible simulated annealing. And simulated annealing, if you anneal it long and slow enough, is sort of going to approximate the global optimum. So what this means is essentially trying to maximize the fitness of this generator. And what that turns out to is that it turns out to finding the smallest weight of this diversity term without having the generator mode collapse. So if the diversity weight gets too small, the whole optimization scheme collapses and the generator only learns to produce one sequence. It sort of fails. For the likelihood cost function, we set that weight to a very large value and then let the likelihood cost function be a margin loss such that we try to approach the mean training set log likelihood. So essentially try to generate sequences that are as the training data. So here I'm going to show highlight results of using deep exploration networks to generate maximally strong polyA signal sequences. And again, the fitness predictor is a parent. And for validation, we're looking at predictions from this independently trained direct APA model. So here we're looking at 4,000 sequences designed by 4 different algorithms. And I'm plotting the predicted fitness scores of these designs. And from left to right, we're comparing these 4 methods, again, trained on a uniform sample of the data, and a feedback GAN trained with a typical parameter setting for the threshold, feedback threshold. And the pink or purple violin corresponds to simulated annealing sequences. And the yellow violin corresponds to the deep exploration network sequences with the red. So as you can see, the simulated annealing method and deep exploration network seems to converge to sequences with very high predicted fitness scores and roughly equal predicted fitness scores. And now if we look at this non-optimized for validation model, we can see that their validation scores are higher than the GAN models while being very, very equal. And furthermore, when we look at the edit distance between sequence samples from these algorithms, we can see that the feedback and simulated annealing and the deep exploration network have roughly the same diversity. So, if you look at these plots, you may of course say, well, so then we should use simulated annealing, right? Because it's marginally better than the deep exploration network. And fine. However, the big upshot with a deep exploration network is that it's considerably faster, right? Because once we have paid the initial cost of training the generator, it's very fast in sampling new sequences. Whereas simulated annealing, you have to essentially run that very long for every sequence you design. And furthermore, with this deep exploration network, it's quite easy to tune this diversity weight or the diversity cost to make more diverse sequences, if you will, of course, at the cost of diminished predictive fitness. And that's the last thing to say. So we also sampled some of the highest predictive fitnesses from this deep exploration network or those sequences, and compare them to our original Seekprop designed sequences in a qPCR assay, where essentially the deep exploration network poly-A signals competed with the Seekprop designed sequences, so the original activation maximization sequences. And on average, we observed an 11.6-fold odds ratio improvement of these dense sequences. So they seem to be even stronger than these Seekprop sequences. Okay, so now finally, let's jump into the third problem, sequence interpretation. And so here I will talk about a method that we recently developed for interpreting sequence-based neural networks. So sequence attribution can be challenging. And to illustrate that, we're going to be switching gears, a 5' UTR sequence at the beginning of an RNA molecule, where we have an in-frame start codon that's marked in red, and an in-frame stop codon that's marked in blue. Together, these two starts and stops create an in-frame upstream open reading frame that represses translation efficiency. Now, luckily, we have a convolutional neural network that's capable of accurately predicting mean ribosome load, or MRL. So it looks at this sequence and predicts low MRL. And in fact, if we run in silico mutagenesis, or ISM, with the CNN model, we can very easily pinpoint these two important features, right? Because they're going to light up in the heat map. Because making a single mutation to either the start codon or the stop codon removes this or destroys this low MRL prediction. Okay, so we can easily interpret the salient features of this sequence. Now watch what happens if I introduce multiple in-frame starts and multiple in-frame stops. We still have a low MRL prediction because we have these open reading frames still. But now we get this kind of bad saturation effect where one start codon essentially against the other start codon, meaning that when we run ISM, any single mutation to one of the starts will have no net effect on the prediction because the other start is there to save the day. So now we have a 5' UTR sequence with a very low predicted MRL, but our interpretation method tells us there aren't any important features in the sequence. And that's, of course, not very good, not a very useful interpretation. And so that's just one example of how nonlinear combinations of feature groups can create challenges for attribution. But there are many other challenges. Just the nuisance of dealing with these discrete sequence patterns can trip up some interpretation methods, for example, those based on gradient saliency. Furthermore, interpreting a single pattern at a time can be quite time-consuming. And finally, if our predictor is quite noisy, our interpretation methods can in some sense overfit to the predictor and highlight features that are for some spurious artifact rather than marking biologically relevant features that are relevant in expectation across many examples. So the solution, in my opinion, is that we should learn input masks that learns to reconstruct the original prediction using as few features in the sequence as possible. So our mask-based approach would choose to keep only one start codon and one stop codon, because that's the minimally sufficient set of features that allows us to reconstruct this low MRL prediction. So that sounds good, but how do we actually find these masks? In fact, there's a rich history of mask-based interpretation methods that most of them originate from the world of computer vision. There, they mask unimportant features in an image by blurring or fading it. Other methods either zero out unimportant features, and other methods replace unimportant features with samples, discrete samples from some background distribution. And that type of masking sounds quite good for biological sequences. There's another axis to this, which is how do you actually optimize these masks? How do you find them? We can either optimize one mask at a time to explain every input pattern, which of course sounds a bit time consuming. Now, the other approach would be to train a parametric model that allows us to infer or generate these masks. And so we essentially developed this method that we call scramblers that combines what we think are the best features from these earlier methods for interpreting biological sequences. So here we replace unimportant features with samples from a background distribution of nucleotides or amino acids. And we, instead of optimizing each mask at a time, we try to train a parametric model that we call the scrambler that very efficiently can predict the masks of new patterns after training. And this work was presented at MLCB last year. So to give you a very quick rundown of how a scrambler works, is that you start with your data that contains these sequences that you want to learn to interpret. You then feed them as input to this trainable neural network that we call the scrambler. The scrambler in turn generates a position weight matrix, or a PWM, from which we draw discrete samples, sequence samples, and feed it to our predictor model. And we then compare the predictions on these samples to the original prediction of the sequence. And the goal here is to train the scrambler to generate a PWM such that only the smallest set of features are kept with high conservation, such that samples from that PWM reconstruct the original prediction. This small set of features in the PWM should correspond to the smallest set of features that allows us to maintain our predictive accuracy. And I'm going to sort of just highlight some cool results I think we got for using this method to interpret protein-protein interactions. So here we have a recurrent neural network that's been trained to predict whether two proteins bind or not, given the two protein sequences as input. And this particular RNN was trained on a very large set of rationally designed coil-coil binders from the Baker lab. Rules of binding specificity consist of these crafted hydrogen bond networks between at the binding interface of two partners. And if we train a scrambler to reconstruct these binding specifications, it indeed recapitulates these known hydrogen bond network positions. So it finds these known determinants of what makes two binders bind specifically to each other, but not to other binders. It also identifies these other important features like hydrophobic residues at the binding interface. And compare the scrambler to other interpretation methods at the task of discovering these hydrogen bond network positions using its importance scores. The scrambler is considerably more accurate at finding those hydrogen bond network positions, which we think is essentially attributable to the fact that we're training this model that learns about hydrogen bond network positions from this quite noisy predictor across many, many examples. Now it warns about an unstable connection again, but that's fine because we're almost done. While we were developing this interpretation method, we also sort of worked on I think, some other useful improvements that allows you to kind of efficiently interpret sequences. one thing that we notice is that in some of these biological processes, there can often be multiple alternate explanations to why you're observing a particular effect or prediction. If you think back to our 5' UTR example, you can choose to explain the low MRL either by one of the starts or the other starts. So by introducing dropout layers at the right positions in the scrambler network, specifically right after the mask that it predicts, we can essentially train a scrambler network that allows us to dynamically query the scrambler after training for alternate possible explanations. So for the 5' UTR example, we can dynamically ask this scrambler of alternate explanations for why we're observing low MRL. Furthermore, in addition to simply reconstructing the original prediction of some sequence, we can train the scrambler to find the smallest set of features that either maximizes or minimizes the prediction. And that actually allows us to decompose a sequence into its positively influencing and negatively influencing sequence motifs. And for APA, this was kind of nice and allowed us to decompose sequences into typical repressive and enhancing motifs. And yep, that's actually all I had. So thank you so much for listening, and I'm happy to take any questions.